So here we are talking about biomolecules. So biomolecules are all organic compounds that are carbon containing, of course, that we get from a living tissue. Water, this is in our living body, in our living tissues in our body. Water is 70 to 90%. Water is not the most abundant biomolecule because water is a chemical, like it's not an organic compound, it's H2, it doesn't have carbon. So it's the most abundant chemical or substance. Then the most abundant biomolecule are your proteins. They are 10 to 15%. Then the nucleic acids 5 to 7%, carbohydrates 3%, lipids 2%, and inorganic ions and compounds 1%. The most abundant element in living body is oxygen. It is 65%, and even in Earth's crust, oxygen is the most abundant. Relative abundance of carbon and hydrogen in living bodies is more than that of the Earth's crust. Basically, we have this table here. This shows the abundance of certain elements in Earth's crust and human body. You see that hydrogen, carbon, oxygen, nitrogen, sulfur are more compared to Earth's crust. But your inorganic ions or elements, sodium, calcium, magnesium, and silicon, are more in Earth's crust. For example, silicon, it's negligible in human body but 27.7% in Earth's crust. This is important. And the most abundant is oxygen. Just remember that oxygen, then carbon, then nitrogen, then hydrogen, the relative, like the abundance in human body. Then we have some constituents, inorganic constituents of living tissues. We have sodium, Na+, K+, Ca2+, Mg2+, H2O, NaCl, CaCO3, phosphate and sulfate ions. Now we are going to chemical analysis of living tissue. To analyze a living tissue, we need to take a living tissue. It's usually a vegetable or piece of liver. Then we are crushing or grinding it in acid. This acid is trichloroacetic acid, CCl3COH, with mortar and pestle. A thick slurry is formed, which is strained with cheesecloth or cotton. We get two components: acid soluble, the ones that are soluble in this acid, which is called filtrate, and the ones that are not soluble in this acid insoluble or retentate, like they are retained. Let's say this is your beaker or something, and this is your cheesecloth, and here you have all the acid and stuff. And when you're adding the living tissue, which is grinded and all, into this acid, usually some part is going down, but some part is retaining. Some part is filtering inside, that is your filtrate. The ones that are retaining here are your retentate. These acid soluble or filtrate, acid soluble pool, usually have your biomicromolecules. Acid insoluble have biomacromolecules, but there's an exception, are lipids. Basically, biomicromolecules have molecular weight of 18 to 800 Dalton, which is less than 1000 Daltons. But in biomacromolecules, the molecular weight is more than or equal to 10,000 Daltons, way more. So these biomacromolecules include amino acids, monosaccharides, disaccharides, nucleotides, fatty acids. Biomacromolecules are proteins which are formed by amino acids, basically chains of amino acids, polysaccharides from monosaccharides, nucleic acid from nucleotide, lipids from fatty acids. Okay. Now, why lipids, even though they are biomicromolecules coming under acid insoluble pool? Lipids, their molecular weights are less than 800 Daltons, but they are acid insoluble. Why? Because usually lipids are found in membranes, last phospholipid